Well, this next presentation is on drug discovery, going through all the different phases of clinical research and trials to the marketplace. The book has some great information about this, and this is really just supplemental material to help from that standpoint of getting everyone in a frame of looking at ethical questions as we go through these specific areas. So to begin with, drug discovery, which I'm going to make the assumption that all of you are aware of the process of drug discovery all the way through to the marketplace. And so we're just going to highlight some aspects of it from that perspective. But to begin with, if you want to think of drug discovery in a very broad sense, it's a way of trying to find new insights into a disease, new insights for researchers to really gain a better perspective of how they can do something from a medication or device standpoint that can either impact the disease state, can stop the disease state, reverse, or slow down. So there's a number of different areas of why we look at drug discovery. And there's a lot of different types of tests that can be done. And this has changed over the years. When I was actually first working in the pharmaceutical industry, this is back in the early 80s, drug discovery then was quite different than it is today. And today, with the advent of new technology, be able to really screen multiple compounds in a very short amount of time, it has really begun to see the process change. However, the ethical questions still exist. And so we need to continue to think about those from that standpoint. And drug discovery can look at a variety of areas, from basically taking a look at finding something, again, new and novel aspects from there, or perhaps something that has already been in the marketplace, and how do we make it better? So there's kind of that differentiation of how we can look at these types of things and see where we can go. There's a lot of specific targeting of molecules that we're looking at from that standpoint. And as I said, while majority of items we talk about in clinical research are related to medication and drug discovery, there is obviously research as well in the device area. And once we get to one segment here regarding clinical trials, and actually when we get to the second part of this specific series for this week, we'll talk a little bit more about some of the research that is done from a standpoint of looking at discovery and looking specifically at devices as well, being used together. So what kind of ethical issues are we really looking at from that standpoint? And the first thing that I would generally begin to look at is what we discussed last week, and looking at specific questions and how to design a research question, and the criteria that are listed there finer, which if you go back and look, you'll see that that's the kind of acronym for the 5 different areas that we looked through. So there's a number of ethical concerns that can really be weighed through the process here. But the question is not necessarily what's happening from a standpoint specifically of drug discovery. The ethical concerns are more of making sure that the process is appropriate, using good clinical practice and things of that nature going through the process. So those are the main things we would look at in this specific section. Another part of this is when we look at drug development itself is the ADME, the absorption, distribution, metabolism, and how it's excreted. And we also begin to see if there's some potential benefits and looking at the mechanism of action, so that we may find a way to deliver the medication differently than what's currently there, and also trying to find a dosage from that standpoint. So, as development continues, we really try to see what's also the best route of administration. Is that orally? Is it by injection? Is it sublingual? What are the different options that we have to take a look at? And we begin to start asking some questions at this specific time frame. Is how does this specific drug development, what we're looking at, affect groups of people? So as an example, is there a difference by gender or race or ethnicity? We know historically that drug development has not been looked at in specific areas. We can look at it by gender, that, for many, many, many years, women were not participants or very minority participants in clinical trials. That has fortunately changed, because I believe that everyone should have the opportunity to participate, and also getting results that would be much more diverse for a population that the medication may be used for, but also beginning to take a look at its effectiveness compared to similar medications that are there. So we have now this gambit of looking at different items and how we can find it and where to go with it and what we can do from that perspective. So we move forward and begin to look at preclinical testing. And we have both in vitro and in vivo. The main issue, of course, that we're looking at here is safety. And with the safety, we're looking at toxicity testing. So now there's a lot of ethical questions that begin to come to fruition. We begin to see the difference of do no harm versus do good. And again, as I explained earlier when we talked about philosophers and things, those do mean different things. Doing good, from that standpoint, you may actually have some aspects of harm involved in doing good for someone, or do no harm would not allow that to happen. And there are many questions about the value of in vitro. And while some do still question that, there is a lot of value. There are ethics that are brought up, in certain aspects of that, but not really that often from that standpoint. However, we begin to see more regulatory impact, and regulatory impact can have issues from an ethical standpoint of looking at the regulations. Are they appropriate? Are they diverse? Do they provide the information that's needed to make sure that there is the aspect of value versus cost? And where do we go through from that perspective? Moving on to the IND phase, we're now looking at something that's very, very different. We're looking at application prior to human testing. We're really taking a look at review of results from preclinical testing, trying to determine side effects and other safety features. As I mentioned in the previous step, it's really looking at safety and toxicity. We can now see that once moved from here and accepted into human trials, and this is where the patent exclusivity is there. And again, the patent exclusivity is changing over time. We'll see what happens as we continue to move forward with this. But ethical issues, again, as we're moving to the next phase, again, the question of do no harm versus do good. What is the risk versus the benefit, value, and cost? And now we can begin to start asking, is this for an individual or is this for the community? And if you want to weigh that a little bit, you can think of it perhaps in this perspective. When you're looking at orphan drugs that may go through different clinical trials, those are for a very limited number of people. So the question is an orphan drug really designed for population health, or is it really designed for a specific disease state and community within that area? And then broken down, is it really designed for the individual patient, because there's such a few number of patients from that perspective. So as the trial begins in phase 1, we're starting to be looking at the dosage range and tolerance and things of that nature. We're looking at 20 to 30 volunteers, for lack of a better term. We're looking at the absorption, distribution, metabolism, and elimination type studies. Sometimes these studies are conducted with those who do have the disease state, and we've seen this with cancer and AIDS and some other areas as well. And the pharmacokinetic trials are usually considered phase 1 as well. And that's regardless of where they start within the overall trial itself. So depending on when it is officially listed in the protocol, many people still consider this to be part of the phase 1 trial. So this is where the ethics issues begin to get addressed. Healthy versus non-healthy participants. I believe we're all in agreement that if we're looking at something for for AIDS or cancer as an example. And there's a possibility that a patient who has either of these disease states could then benefit from even this small aspect of the trial, then we want to make sure that they can get involved. Healthy volunteers, 20 to 30 from that standpoint. It's always interesting to me because we do see many times at major research universities. So I would explain that Campbell is not a major research university from a standpoint of clinical trials. But if you look locally at Duke or UNC, East Carolina, those areas that have medical schools, those are the ones we would look at. And the reason I bring that up is that there are many times where we see, phase 1 healthy volunteers as actually being college students. And part of that's because of the payment for participation. Now, most times in this area, it's fairly minimal payment, but there is a payment. The question that comes up as part of the discussion board is the new aspect of a right to try. The FDA already has a process in place for a patient to submit information for them to gain access to a medication that's in a trial. But the newer proposal is that anyone could ask for this right to try. And from an ethical standpoint, that would have a huge impact on clinical trials. We try to make sure that we are looking at a specific methodology, a specific protocol. And when you go outside of that, that impacts data and results and also impacts the ethics, because now we're going to have to take a look at, besides the criteria that we set up for inclusion in the study, we now have to probably yes, design another set of criteria for those who want to go through the right to try. Because we don't want to get someone in, even on a right to try, who's not truly eligible for this specific product that we're looking at. So this is going to add some tremendous ethical concerns. I don't think they've been raised at all in the legislation and things that have been moving forward, but I find this to be a very, very concerning aspect for me personally. We move on to phase 2, and we talk about 2A and B, depending on how people look at this, but we're talking about now looking at trials to evaluate the efficacy and safety in selected populations, 100 to 300 patients who have the disease state or condition. Can involve hospitalization of patients to be closely monitored. And I'll give you the example here. So if we have patients, and I'll use the mental health field as the example, let's say we're doing a study for bipolar. And part of the issue is that we want to make sure that the patients are clear of any other medication they may have been on, or whatever the criteria is set up as. We may want to have them closely monitored either in a hospital or some type of a setting where we control as many environments as possible to see how the medication is actually doing this. I bring this up because if you go down to the ethical issues, the payment for participation there is much different than someone who is asked to travel to come in once for something and 3 weeks later and then 6 weeks later. Those are completely different aspects of a trial. And we need to really understand that process itself. Is it truly beneficial for a patient who's testing a product that's in very early stages to be asked to stay in an area? Basically, everything is controlled from that standpoint, because once this moves to the next phase, we move out broader, it's not in that same kind of environment. So are we creating an environment for those specific studies that may not really provide the results that we want for the general population? And then B is trials to evaluate safety and efficacy in patients who have the disease condition to be treated. They usually represent the most rigorous of a medicine's efficacy. Sometimes they're called the pivotal trial. This is where a lot of the go, no-go decisions are made from that standpoint. But from an ethical standpoint, again, we'd want to take a look at how we have the full disclosure and informed consent, so people fully understand what the aspects are here. And we'll go through these later, but the idea of saying, is it written at the appropriate level? Does a person truly understand? How do we test to make sure the patient truly understands? A variety of items that we'll get into. Have there been the appropriate inclusion and exclusion factors that are involved with the recruitment of specific patients? And do the actual investigators have the knowledge of what they're looking at, or are they making assumptions of what will happen and trying to predict what they're looking for? Those may not sound like a concern, but in actuality they are. In some areas, we are not going to have the knowledge. In a brand new therapeutic area where we have no additional products to look back at, we have no trials we can look back at, the knowledge just isn't there. We can do as much as we can to find out as much as we can. But when we are in a therapeutic area where we've had other molecules that have moved forward into the marketplace, then we do, and we want to make sure that everyone who's involved has that knowledge and they're not making assumptions, that they're really in that area of understanding fully what is happening. And also from the patient perspective, I think that's perhaps one thing that may be missing in some of these areas as we talk about them. All these aspects we'll talk about, from the investigators to recruiters to everything else. They can have all the knowledge and everything they want from their own perspective and their own background, but they definitely should be able to put themselves in the position of the patient who may not have the understanding of, or the health literacy to be able to understand specific items that are there. In phase 3, we begin now to look much broader, multi-center studies, 1,000 to 3,000 patients. Now we're looking at additional safety and efficacy data. And before I mentioned the idea of having this in controlled environment, now we're looking at controlled and uncontrolled designs. We're seeing all different types of ways to do this. Looking in special groups of patients, special conditions. All these aspects are based on how the protocol is written. And in B, again, it can be after submission of an NDA from that standpoint. And so we're really looking at the period between submission and approval or receipt of marketing authorization. And I put down the ethical items here, and it says many, because we're now expanded to 3,000 or sometimes many more than that. We now do run the gambit of ethical concerns, making sure that the diversity of the patient population, making sure that the informed consent is written at the appropriate level. I think the book does a good perspective when you go through reading these, defining what's happening here and giving you the opportunity to really think through what ethical questions would you want to ask, going through that aspect. And then, phase 4, some people consider this to be post-marketing surveillance. Others consider that to be a phase 5. It really depends on your terminology and how you look at this, but, it's really evaluating the formulations of dosage, duration of treatment, other interactions with other medicines, any other factors that may come in from that standpoint that are basically post-marketing. It's observational more than experimental. And so it's a different perspective here. But most questions from an ethical standpoint should have been answered before we get to phase 4. Because at this point in time, we're now out to everybody. But here's the question that I hope you guys will think through from a standpoint of understanding that while we may ask everything we can still run into issues. So in the last 10 years, we've had medications that have been approved, gone to market, and then they have been pulled off the market because of post-marketing surveillance information that has shown greater toxicity or has shown aspects of side effects that were not being picked up in the other trials, whatever it may be. And so this means that the ethical impact is very high, because if you've given people medication that has gone through this process, and now you decide that no one should have this anymore because of items you were finding after the clinical trial, in a sense, has been completed for phases 1, 2, and 3, that's a very large ethical concern. That means somewhere in the process, we did not gain enough information. There was something in the protocol that did not look at different aspects that were then found later after it reached the marketplace. So a very high ethical impact when we're looking at that kind of information. And as I mentioned, from a clinical trial standpoint, some people look at this as post-marketing. And this is new data shared with others. And as I just mentioned about the concern of items that you pulled back, there's also new data that comes out and shared. But the question is, when is it shared and how is it shared? We're very much well aware of the fact that we have products that have gone through all this, come out for a specific disease state, and all of a sudden we find a side effect, where someone who's taking an antidepressant all of a sudden has smoking cessation at the same time. So new data comes out, when should it be presented? As soon as information is beginning to be seen in the data, and how, it's a big question. Again, I'm making the assumption most of you are aware of the fact when you look at clinical trials, today we can find things online, which is great. But when you look at publications still, the publications of the trials or the publications of the studies, they tend to be the publications that are positive, that have had some kind of a positive outcome. We rarely see the publications of those trials that were stopped at any phase, the reasons why they were stopped, and other aspects. So we still have a long way to go as far as when and how information is provided. So let me give you a question to ponder. If aspirin were moved to a clinical trial today, would it be approved in an IRB? So just think of aspirin and what it does today. What is the risk versus benefit? What is the value versus the cost? Aspirin today is very inexpensive. If it were to go through a clinical trial as a brand new product, it would not be inexpensive based on the cost that we have for estimates of what it takes to reach the market today. And risk versus benefit is intriguing because aspirin, while extremely effective in what it does and how it works, does have a large number of side effects. And the question is, would those side effects actually prohibit it from moving forward in a true clinical trial today? So I leave you with that, just an example to think about. I hope this has been informative, and looking forward to providing more information and reading more in our discussion board. And lastly, for this week, let me apologize. I had sent out a note to everybody. I had been traveling last week and was not accessing my email, which, as I noted in the announcement, is truly unacceptable on my part. So I apologize to everybody. I will be getting back to everybody this week with questions you have written in and other items. My travel schedule is very limited moving forward, so I should not have that issue again. I thank you again and look forward to seeing everyone online and getting information. Thank you so much. Have a great day.